here. I haven't been here for a while, and you're only like 3 hours away. Actually, 2 if I'm in my Mini. But I shouldn't admit to that, should I? Well, 2 and 20. But it is really nice, and it's also really nice to be part of the Rough Cut seminar series because that's kind of a unique seminar series. It's bringing people from Montana together to hear about what we're all doing. So it's really nice to be included in that. Today I'm going to give a talk titled A Tale of a Beetle and Two Fungi, How a Partnership Built an Empire and Led to Its Demise. It's a very dramatic title. I think it's actually a pretty dramatic talk, at least the topic. These beetles are pretty interesting. The beetle, of course, is mountain pine beetle, which you've all heard of, I'm sure. The fungi are two symbionts of this beetle, which you may or may not know about. Kind of where I got this idea of empire building by a beetle was the title of a book that was published a couple of years ago by a guy named Andrew Nikiforuk. And in this book, he kind of chronicled this big, massive outbreak this beetle has developed over the last few years. And it really is kind of a story of empire building, because this outbreak is really amazing in its extent. This outbreak happens to be something like 10 times bigger than any this beetle has ever done before that we know of. Okay, and it very well may be actually the largest insect outbreak that's ever been recorded by humans on the planet. So it's a pretty major thing. But one thing that kind of stands out also is that this incredibly severe outbreak hasn't been restricted to the natural range of this insect. This insect, because of warming, has actually moved hundreds of kilometers further north. In fact, it's all the way now to the Yukon. It's jumped the northern Rockies and has spread across Alberta. It's now into Saskatchewan. And it's in the boreal forest, which is made up of a new host tree species for this insect called jack pine. So in this area of Canada, the beetle is actually acting essentially as an exotic. And for those of you, and I know Kathy knows this very well, but because of warming, the beetle has also been able to move up in elevation, and it's now causing a lot of devastation in whitebark pine. So it really is sort of a tale of empire building by this little insect. And it is pretty remarkable that this insect could do such a thing. In Andrew's book, he likens it to something the size and appearance of a mouse turd, which I thought was really good. So I went out and found a mouse turd. Hey, that's perfect. This was the mouse turd, by the way. But if you think about it, it is amazing that something this tiny can have done so much that we've seen. These seas of red, these pictures we've seen, literally millions and millions and millions of hectares of forest that are dead. So it is pretty remarkable. And you might wonder, how does a little insect the size and appearance of a mouse turd do this? Well, it's actually not easy for the beetle to do. Most of the time, the beetle is present in really low numbers. It's rare, it's hard to find, and at those times, it's actually at the mercy of its own low numbers, because this beetle has to mass attack trees to kill them, to overcome their defenses, and if not enough beetles show up for the attack, the tree wins and the beetles die. And it literally takes hundreds or thousands of beetles to kill a tree. So if you only have a few beetles out there, when they initiate attack, very seldom are they successful. So they hang on in these relatively rare, weak, stressed trees, and they really struggle. Not that anybody here worries about them too much. But they will hang on in these low numbers for literally decades, sometimes even centuries, until you get something we call a trigger.
Is it inoculum dose have to be that high in order to overcome a tree defense, or does it take that many beetles to
It takes that many
predation?
beetles. Takes that many beetles to knock the defenses down. Yeah, and I'll get to that. So we get a trigger, and this is what actually allows the beetles to expand their numbers. And the trigger for a mountain pine beetle is warm and dry. So warm temperatures actually allow greater productivity of the beetles and greater survival, and of course that means you end up with more beetles. Dry means you have drought-stressed trees. And drought-stressed trees don't tend to have as good defenses, so it doesn't take as many beetles to kill them. And something else that's really important about drought is drought doesn't just stress trees in little localized areas typically. It's usually a region-wide event. And so we're talking a lot of trees across the landscape that become easier for the beetles to kill. So the beetles begin to build up, and then they finally reach over a threshold over which there's so many beetles out there that they can actually then kill healthy defensive trees. And at that point, something amazing sort of happens, and that is that they begin to actually prefer healthy trees. And that's maybe not surprising, because healthy trees are a lot better food for the young, and that results actually in a lot more beetles. And so you really, at this point, have the outbreak kind of off and running. And there's really nothing you can do about it at this point. And the outbreak will end up in a couple of, one or two possible endpoints. One is they can pretty much kill all the susceptible hosts in an area, and then they kind of crash out because of a lack of resources. The other option is that you may have a return back to cooler, wetter conditions, and then you have lower productivity, lower survival. Trees begin to build up their defenses again because they're getting enough rain, and the whole population sort of begins to decline. Okay? So it actually takes a number of things to coincide in order to get an outbreak. Now, it turns out there's actually a whole lot more to building an empire than just being able to kill those trees, okay? So the beetles have gone through all this work and danger, literally, because this is a very dangerous thing for them to do, to go into a tree. And then when they get in, they kill the tree. Oh, now what? You're in wood. Wood is like, you know, almost a non-food. There's very little nutritional substrate or substance in wood. And so how do they deal with that? Well, what they've done is that they've partnered with a couple of fungi, Ophiostoma montium and Grossmania clavidra. And if you look at trees that have been killed by the beetle, you'll see this really pretty blue stain in the wood. Some people call it denim pine and make some nice furniture and stuff out of it. The staining is from the fungi, okay? And these fungi do something pretty amazing for the beetle. They provide to the beetle nutrients that allow them to survive at an otherwise unsuitable food source. So they provide nitrogen, and they provide sterols that allows them to grow, produce hormones, reproduce, and so forth. But the fungi, while they both provide these similar benefits to the beetle, aren't equal. And you really wouldn't expect them to be. They're different organisms. Turns out that Grossmania clavidra is by far a better symbiont. When the beetles develop with that fungus, they're bigger, they live longer, they can fly further, they have more eggs, and so they have a real fitness advantage. But while Montium is sort of just okay, it's still by far much better for the beetle than no fungus, because without the fungus, or either of the fungi, the beetle can't survive. There's another important difference with these fungi. Okay? It turns out that Grossmania clavidra, the good fungus, likes it cool, and Montium, which is the warm-loving fungus, likes it warm. And this will be a really important point in a little bit. Okay, so I want to take you through a life cycle of the beetle and the fungi and show how these are really intimately linked through time. And I'll start you up here with this little beetle. She has just left the tree that she grew up in. Now she's got to go off and choose another tree to attack and have her kids. If she's the first one onto a tree, we call her a pioneer beetle. And for her, she has to do all the host choice stuff. Is this the right species? Is it the right diameter of tree? Is it the right defensiveness? Is this one going to be too defensive? It's got to make a lot of decisions. She'll somehow, we haven't worked this all out, figure out this is a good tree. She'll enter and release an aggregation pheromone. And that will draw in lots and lots, she hopes, beetles to the tree. And if she's lucky enough to get enough help to show up over just a few days, then they will knock down the physiological defenses of that tree, and then they can get in under the bark and begin to lay eggs. So let's say she got lucky and enough help showed up, they overwhelm the defenses. At that point, the tree won't recover, They get under the bark and they produce these kind of galleries, and along the edge they lay the eggs. And as they do, as they construct these galleries, they also inoculate the tree with these two fungi that they're carrying. The eggs will hatch, and the little larvae will tunnel out, and you can also see this sort of blue stain forming where the fungi are starting to grow into the tree. And as these larvae tunnel during their whole development, they feed not just on the phloem, but also on the fungi, and that allows them to complete development. Now these fungi are not trapped in the phloem like the beetles. The beetles have to spend their whole development period in the phloem. But the fungi can actually go deep into the sapwood, and there they do something pretty remarkable. And that is that they gather up the nitrogen that's there and somehow move it back to the area where the larvae are feeding. And they actually increase the nitrogen by something like 40%. And if you're a nitrogen-limited insect, 40% is astronomical. This is big. So that's a pretty big impact. Then, over the year-long period of the beetle, they continue to develop. And then eventually the beetle becomes an adult, but they don't leave the tree right away. They stay in there, and hopefully you can see this. These pupal chambers become lined with spores of the fungi, and as these new teneral adults, they stay in there and feed on these fungal layers, and that serves two functions. One is, if we found that if they aren't allowed to feed on these spores, they don't reproduce. So that's a pretty big function. The other one is that as new adults, this spore feeding is what allows them to pack these little fungal suitcases they have in their mouth parts called mycangia. So they pack these full of spores so that when they leave the tree, that ensures the fungi go to the next tree and the next generation of beetles. So up here is actually a scanning electron micrograph of a real intense close-up of these little pockets. This is the opening, and these are the fungi extruding right out of the opening. These are pretty remarkable little structures. They're highly specific. They only pick up these two fungi. And by the time this tree's been dead for a year, there's all sorts of other stuff in there, but none of it gets picked up. And also, they appear to produce secretions that nurture the growth of the fungi, because these things do grow right out of the opening. And so they do appear to be very highly evolved. So these are really closely linked, and it turns out it's a very, very old association. So something I wondered about with this symbiosis, but also a number of others, because it's not just mountain pine beetle that has fungal mutualists. A lot of other bark beetles do too, is why have two fungi? And it turns out that's a consistent pattern with most of these beetles that have mutualists, is they don't have one, they've got two. So why have two? If you've got fungi that, one is way better than the other one, why put up with one that's just sort of okay? You would expect natural selection to move towards the one that supports the highest fitness, but we don't see that. So I've thought about this for a lot of years, and some of the things we've come up with are ideas like, well, maybe it's good to have redundancy in the system. If you're completely dependent on another organism, something happens to it, you're toast. So if you have two, you may, have some redundancy in a backup system. But also, looking at these symbioses, and again, not just mountain pine beetle, but a bunch of them, they always seem to have two, and they have different temperature tolerances. So maybe, whoops, I'm going the wrong way. So maybe by having two, like mountain pine beetle, one that likes it warm and one that likes it cool, you might be able to increase your ecological amplitude. You may be able to live in a more variable habitat, because if it gets warm, you've got one. If it gets cool, you've got one, and you're not in trouble. You're never found without a fungus. So this may be the case. But it's one thing for having a multipartite system being good for the beetle. There are some real complications with trying to maintain the stability of such a symbiosis in nature. And one of those is that these fungal symbionts are expected to be highly competitive with one another. So if you think about it, these fungi get inoculated into the tree at the same time. They have to live in the same space. What we've found from some studies we've done is they actually go after exactly the same kind of easy sugars and amino acids. And at the end of a year, they've got to compete for the same host beetles leaving the tree to get transported. So you'd expect these to be competitive, and you would expect whichever one is the most highly competitive to dominate over time, and you'd move to a one symbiont, one host system. But like many of human predictions were wrong, because obviously that's not a problem here, because the symbiosis has been stable for a very long time. So there's got to be some sort of mechanism there. So our idea is that the mechanism is something like temperature variability, really. That these fungi have different tolerances to temperature, and so they fluctuate with the environment, and no one ever gets to dominate. Okay? We have some evidence that that might be the case. These are very highly temperature-driven systems. What we found is that the relative prevalence of these two fungi with the beetles over time is completely temperature-driven. When it's cool, the cool one dominates. When it's warm, the warm one dominates, and that can shift very rapidly. So for example, this is a graph from some older work. What we did in this study was to go out and collect beetles over a 3-month period at a whole bunch of sites across 3 states, warm sites, cool sites, intermediate sites, and collect the fungi from beetles that were dispersing and look to see how that related to temperature. And for this graph, it just shows that as you have more and more days over 32 degrees C, what happens is beetles carrying the cool fungus just crash out. It doesn't get carried anymore. And there's a complete switch over to beetles carrying the warm fungus. And under cool situations, you get the complete opposite. So 1 week, you might have one fungus go, temperature changes, the other one gets to go. It's that dynamic. And we also find that kind of a shift occurring in the year-long period of development in a tree. So in cooler parts of the year, the cool one dominates, and then it loses ground to the warm one later. So we know these systems are temperature-driven. So our question was, can temperature actually stabilize these systems? And so to get at this, we needed to develop a couple of models, temperature-driven models for the fungi. And once we had the models, what we wanted to do is see if intra-year variability could stabilize the system by itself, inter-year variability because different years are different, and because we know beetles don't stay in the same place forever. They move upslope, they move downslope, they get blown over into the next valley. We added a scenario where we migrated them between different thermal habitats. And to put some biological realism into the models, we also had to recognize that these fungi don't live in the tree by themselves. They're living in there with other individuals. They're living there with other individuals of the same species and other individuals of the other species. And something kind of cool about bark beetles is that they attack trees in a fairly regular spacing, okay? And so we can use this spacing that they use to attack and count that as a nice proxy for inoculation, because everywhere a beetle bores in is where the fungus gets inoculated. And we also know that in nature, the density of these attacks varies by the background population of your beetles, and so we could also play with the density to see if that has an impact. So to develop the models, we had to go out and get a lot of data. So we had to isolate fungi from a lot of populations, so we worked with a wide range of isolates, something like 80, so that we could not only get really good growth curves to run the models, but also a really good indication of the amount of genetic variability that's present in these species. And that was really important for us to start looking at the effects, potential effects of climate change and how these fungi might be able to respond. We took those and grew them at a lot of different temperatures, billions of plates when you've got this many isolates, kept track of their growth rates. Then we had to see how they interacted together in the tree. Something nice about these fungi is that they're incredibly well behaved. Most fungi, you put them in a plate, you've got this vicious battle going on. I mean, it's physical and chemical warfare. These guys just grow together and stop. It's a nice classic case of scramble competition. Makes it a whole lot easier to model as well. So we had to figure that out, how they competed with themselves and with others, the other species. And very importantly, we had to know what temperature these things sporulated at. Because we could model the capture of space in a tree, but if they're not sporulating at exactly that point those beetles are leaving the tree, it's a dead end. They're not going anywhere. So we had to know that too. Well, of course, we had to do field validation. For one thing, we had to go out and figure out how they actually grew in a tree, because no surprise, they grow different in the lab. They're a little faster. So we found out how they grew in a tree so we could scale that for the model. And what else? Oh, yeah. We also had to do some field validation, see what happens in nature so that we could tell if our models were actually coming out the way they should. So we went out to a couple of sites, and one of them for 2 years. We probed 5 trees at each of those sites with probes into the phloem, took temperature measurements every 15 minutes. At the point of attack on those trees, we kept track of, we started trapping beetles from the area to see what proportion of the fungi were being carried by that population so we knew what was going into the tree. Then for the several days of the attack, we sat by the trees, and every day we marked the new attacks with colored pins. So one color one day, the next day all the new attacks, new attacks got another color, next day and new attacks with another color. Really pretty tree at the end, was damn boring to do. But so we did that for several days, and then periodically over the year, we removed some bark, found out what stage the beetles were at, did grid isolation of fungi to figure out their dominance. And at the end of the year, we caged the trees, collected beetles, and we knew the sequence they were coming out, and we also isolated their fungi so we know what came out with them. And so we used that to see how accurate our models were. So we developed the models, and I'm not going to present those because when I say we, it was actually Jim Powell, who was part of this project at Utah State, and his grad student Audrey Addison. I can't really describe mathematical models. I'm more the ecologist on this. But we developed those. We coupled them with a very well-validated phenology model for the beetle, and then we ran those with 2 data sets. And these are really cool data sets because they're from trees at a mid-elevation site and one at a high-elevation site for 10 years straight. And we could use those. So I'm going to tell you what we found. I'll walk you through these graphs. These are our results for, let me back up. Our sites were different. Again, Stanley Valley was mid-elevation, okay, typical lodgepole pine for mountain pine beetle. And then Railroad Ridge was up in the whitebark pine, which is a pretty cool site, much higher elevation. So we have a kind of a warm, cool site and then a cold site. So this is for the mid-elevation Stanley site, and what we did here, the lines are where we took a particular year of the 10-year data set, and then we just stacked it on end for 200 years. And each of these boxes is a different attack density, and this is actually the range that you might expect to see out in nature. So up here we have an attack density of 40 millimeters. These are very close together. This is really the high end. This is extreme competition for the beetle, probably peak outbreak. You don't see this very often. And then these are 5 millimeters increasingly apart until you get down here to 85 millimeters apart, which is on the low end. This is a tree where there's so few attacks that it's kind of iffy whether they actually were able to kill the tree. The ones to keep your eyes on in this and subsequent graphs are this one and that one and this one. So, 55 to 65. That's the density you tend to see out there and what is most optimal for the beetle. So we entered the beetles into the trees and the models carrying 50-50 of the 2 fungi. And so if it stays flat, that means we found stability, which is what we're looking for. If it drifts up, that means we're going to move towards domination over time of the good, cool-loving fungus. If it drifts down, that means you're going to move over time to the loss of that good fungus and the domination by the okay fungus. All right? So what you're going to see here is at the very high densities, and again, these are pretty unnatural. You don't find them very often. We got stability. But if we get down to more realistic situations, we see that this is not stable. that over time, given at this site, we would lose the cool-loving good fungus, okay? And the other one would dominate. And the further they get apart, the worse it gets, except for these 2 years, which turned out to be the kind of abnormally cool ones in that particular set. So kind of the take-home message here is that just by itself, inter-year variability isn't going to stabilize the system. So we looked at inter-year, and this is, you're going to have to look at these a little bit different. So here we ran the 10-year data sets over time, but each of these lines is a different density. This is for Stanley, the mid-elevation site, and that is for Railroad Ridge, the high-elevation site, so the cool one. And depending if you're in a warmer or cooler site, you get different conditions. That shouldn't be too surprising. Again, in both, at the very high densities, you have stability. But if you get to the more realistic ones, over time, at the warmer site, you're going to lose the good fungus, and over time at the cool site, that one's going to go to domination. So it depends where you're at, what would happen, but again, we don't really have stability. So we decided to move them around a little because bark beetles are not going to stay in the same place for 200 years. That's pretty not very reasonable. So what we've done here is migrate them between different the two habitats. In this case, we let them stay in beautiful Stanley. If you've ever been there, it's a nice place. We let them stay there for 6 years, and then we moved them up to Railroad Ridge in the cold for 12, and then back to 6, and up to 12 for 200 years, so back and forth. And what we found is that is an incredibly stable situation, except at that real broad spacing that's probably not even a successful attack. So this was really exciting because, you know, hey, this could account for stability. But of course with models, then you get to play and you can change things. So over here, what we did is we only let them stay in Stanley for 4, and we made them stay out in the cool for 20. And over time, as you might suspect, because they're spending so much time in the cool place, you do drift towards the cool, good, cool-loving fungus. I'll get that. But we really do think that we've stumbled on a really strong mechanism to maintain stability in these symbioses, because they do move around, parts of populations move around, and that could really keep either one of these from going to dominance. One of our big questions in developing this project and starting to work on it was wondering what would happen to these symbioses in a warming climate. So what happens if we warm things up a little bit? And with models, you can kind of do that. So what we did was we ran a thermal migration model, but we warmed up both of the data sets by 1 degree Celsius. So not a lot, and in a lot of places we've actually already done that. And this is what we get. It's a pretty dramatic change. Okay? Again, at really high densities, no problem. But if you, and this is at 6 generations warm and 20 in the cool, so this is a fairly cool run, you are going to lose the good fungus over time. This is a pretty dramatic change. Okay? So something to think about when you're looking at this also is that Stanley actually is not that warm of a site. It's a cool site. And if we go out to our warm sites that we work at, our lower elevation sites, some that get very hot in the summer, what we're already seeing is that you may only pick up maybe 1% or less of the population that's carrying the good fungus. That warm-loving fungus is highly dominant when you move to warm sites. And something else we've noticed at some of our sites in Montana, we're getting summers in some places while the beetles are dispersing that are, 95, 100 degrees out. Beetles are not carrying fungi. So when it gets really hot, nobody's sporulating in the tree, and beetles are dispersing without their symbionts. So what we think is that as we have continual warming, in warmer parts of the range, we may start seeing a loss of the good fungus, and that means you're going to have a loss of redundancy in the symbiotic system. But also we might see in some cases the loss of both fungi, and that would completely decouple the symbiosis. And right now, with all the models that are being developed to kind of predict range expansions and contractions for the beetles, they're only considering beetle thermal tolerances. So it might be important, and we're thinking about trying to do this, to look at how potential symbiont loss may be an important factor to consider in figuring out where range contractions occur. I can't find the good fungus in Arizona at all, but it's very hard to find the beetle there because it doesn't outbreak and it's very rare. But I think that maybe says something too, but maybe that's just me thinking my fungi do more than they do. But I think it's going to be interesting to start playing with these models in a climate change context. Now this kind of brings up the idea of risk in symbiosis. And at this point, I want to switch gears a little bit and talk about a study that John McCutcheon at and I are embarking on. We've been working on this for a little bit now, looking at risk in symbiosis. And for most people, when they consider this, they think mainly just the host. So the poor little mountain pine beetle is going to lose its fungi, or corals are going to lose their zooxanthellae bleach. I mean, these are the things that are happening. But mostly people think of it in the host context. But we have to realize it also means risk for the symbionts. And so that's what John and I are looking at. So we're focusing still on insect microbial mutualisms, and that's where both partners benefit. And we know that these things are good. Okay, that's why organisms are involved in them. Insect microbial mutualisms, in fact, have been one of the major drivers in the success and diversity of insects. They're, after all, the most diverse animal group we have on the planet. And there's a very good reason for that, because if an insect partners with a microbe, they do something amazing. They can access genomic variation instantaneously. They don't have to spend millennia, I've got to quit saying this, trying to evolve a trait. That's not the way you put it. They don't have to take millennia evolving a trait that will be beneficial, or even many traits that might be beneficial or something that they're incapable of evolving. But by partnering with a microbe, they can do those traits, those functions immediately. And that has been a very powerful thing for insects. It's allowed them to move into a lot of adaptive zones, and some of the most amazing adaptive radiations we've seen have occurred after these partnerships have formed. And also, it, obviously gives them a great competitive advantage. And it's also good for the symbionts. I mean, they get transported around, they gain functions back from the hosts, but it also leads to a lot of risk for those symbionts. This is from a paper I stole from John, but since he's my partner in this, I think that's okay. But he's done some really remarkable work looking at what it means to become more and more intimate with your host. So what happens as you become increasingly dependent upon your host? And what he's done is work with bacteria that are endosymbionts of a number of insects. And this is an example of the consequences of what it means to be a bacterial endosymbiont with an insect. This genome you see on the outside is of a mycoplasma species of a bacterium. It is the smallest known genome of a free-living bacterium. So it's free-living, it has all the stuff it needs to contend with the real world, but is by no means a big genome. It's very small. But look at the genomes that are held within here, how tiny they are. Those are bacterial endosymbiont genomes. Some are so tiny you'd hardly even think you could call that a genome anymore. So what happens when they become more dependent on their hosts is they start dumping huge portions of their genomes. You might wonder, how do you do that? Well, they're living deep inside an insect. They're getting a lot of functions from their host. And so they can lose things because they don't have to really live in the real world anymore. They don't have to have those functions. And the host provides to them many things. And some of these, like this one, is so reduced, all they have are a few genes left that allow them to encode for things that the host needs. So you can imagine that if something happened to their host or the environment changed so that it wouldn't support the host, none of these guys could revert back to the free-living stage. They don't have the equipment to do it anymore. And they probably can't switch hosts because what genes they have left have been so fine-tuned to the host. So they're actually quite at risk. So what we want to do is look at whether increasing symbiotic intimacy has this kind of an impact on beetle-associated fungi. These fungi are very different because they're not bacteria, they're not prokaryotes, they're eukaryotes, so that's a biggie. And they're also ectosymbionts. They're not living inside the insect, so they still have to deal with the real world to a degree, so they have to keep some function. But it's a great system to do this because we can compare different feeding types that have different degrees of intimacy with the host. So we have bark beetles that actually don't rely on fungi for nutrition. How do they do that? Well, they just have to eat a huge amount of tree tissues. They feed and feed and feed and feed till they can finally become an adult. So they don't depend on fungi. Then we have these mycophloriophagous beetles like mountain pine beetle that don't feed on that much phloem, but they get their nutrition from the fungi. And these are very specific obligate associations. Then we have this whole group that's taken it another step further, the ambrosia beetles. And they actually just dive right into the sapwood, which is basically non-food. And there they grow fungi, and the fungi are their complete food source. So these beetles are all in the same subfamily of weevils, so we can compare among related organisms. But even better, we have two groups of fungi that are associated with each of these. So we have these kind of free-living hitchhiker incidental ones, members of both groups of fungi that ride around on the free-living beetles. We have members of both groups that are with mycophloriophagous bark beetles, and we have members of both groups that are with ambrosia beetles. So we can see if, as these groups get more and more intimate with the host, if they change in the same way or a very different manner. It gets better. This is a phylogeny you don't have to read that was developed by Farrell a number of years ago of the scoletine beetles, which these are in. And what I've done is overlain on this some arrows that show independent origins of these fungal feeding types. So for the bark beetles that feed on phloem and fungi, it's evolved at least 3 times independently. And for the ambrosia beetles, which feed only on fungi now, that has evolved independently. Actually, there should be more arrows, about 11 times that we know of. So we can, again, do comparative work. We can look at these sets. All of these have come out of free-living beetles, so we can compare, in each case, what has symbiotic intimacy meant for these groups. Does it always mean a loss of genomes, or is it other effects? But we can look at these in a lot of different ways. So these are our main questions. We've got a good start thanks to the help with the IOE. We've got 13 genomes now to work with. We're going to add 20 or 30 more. And we're going to start looking to see what effect the potential symbiotic intimacy may have on these genomes. Does it have the same effects? Does symbiosis have the same effects as with prokaryotic endosymbioses, or is it something completely different? And also, something that's a very important part of this is, does genomic change due to symbiosis put these systems at risk? Okay, so under climate change, are they more at risk? And also something we want to look at is another type of rapid environmental change is what happens when you get moved into a novel environment or a novel host tree species. These are the commonest insects moved around and established in forests worldwide. And so it'll be interesting to see what it means when your genome has been altered for that. We have some indications that there is some change going on because the free-living fungi are highly sexual and they're fairly complex morphologically. But as you move towards the ambrosial stage, you have increasing loss of sex and you have a decrease in morphological complexity. And also as you move to become an ambrosial fungus, you go from growing on just about anything, happy in a petri dish, to being very hard to keeping alive. So something's happening. It'll be interesting to see what that is. So with that, I will acknowledge my partners in crime at UM on the project I just talked about, John McCutcheon and his grad student Dan Vanderpool, and we've been supported through the Montana Institute on Ecosystems. And then the first work with the models was funded through NSF, and that was with one of my grad students, Melissa Moore, and Audrey Addison and Jim Powell at Utah State, and Barb Betts at the Forest Sciences Lab in Logan. Thank you.
We have lots of time for questions.
I think I talked fast. I had a lot of coffee. Yep.
You said a couple of times that that association seems to be really old.
Yeah.
How much do we know about that?
We can go probably safely say at least 100 million. Well, there's this general one with these fungi with that genus of beetles. Something we'd like to fine tune. We can do really soon. But it's old. That's why, you know, it'd be interesting to see how something stays stable that long. Yeah, and across such a broad geographic range. I mean, this thing's from the Yukon now to Mexico.
So 100 million years old would be almost the evolution of pine. Pinaceae, right?
Well, the Dendroctonus radiated with a lot of that, I believe, and I may be really stepping out there on this estimate. I better John would say that. But that's when Dendroctonus radiated, and it appears that a lot of these associations formed that far back. And in fact, with some of the population genetic stuff we're doing, the differentiation among populations is consistent.
Do you know what the relationship is affecting the reproduction of the insects at all? Are there?
We would suspect so, because when the beetle develops with the OK fungus, productivity is reduced by about 35–40% in the lab. We expect that would probably translate to the field. We'd like to look at that. It's kind of hard. I think with some qPCR we could figure that apart, because the fungi look the same in the tree. They both blue stain it. You don't know which one's there. But if you know exactly what's there, you could track which fungus the beetle is developed with. We used to try to get at that by cutting trees down, putting the chunks in rearing containers, and the ones that came out, we isolated the fungi and measured the beetle and then assumed that what the beetle was carrying out was what it fed on in the tree. And what we found is, for the most part, those that fed on that were carrying the good fungus were always a lot bigger, you know, in most studies. But now that we know the fungi kind of give and take space in the tree over time, we're not so confident that what they're carrying when they come out is what they fed on as a kid. So I think we're going to have to get a little more precise with that. But I would expect some pretty big impacts on population dynamics, because they are pretty different in their effects. Mm-hmm.
So you said that the beetles that are flying, emerging at around 100 degrees are not carrying fungi. Is that because of a sharper temperature increase in the spring? I mean, isn't there emergence Q temperature?
Sort of. It's not a Q. It's more when they there's the combination of, you know, temperature-driven development. So depending what stage they are, when it warms up, they'll begin to develop. And they do tend to like to fly at a certain temperature, but they have to develop at a certain rate to get there, and that's pretty staggered depending on.
But they would lose the fungus at emergence.
If it gets really hot out early, or actually at some elevation, you know, they only come out like late July or early August, so if it's really hot, all of a sudden they won't have any fungi. But what we think is that it means that sporulation has been shut down. It's too hot for the fungi. Fungi don't typically like hot. I don't know, Kathy, what do you think? Would that be a good?
That's possible.
I think that's probably,
Yep.
I mean, fungi don't like heat. It's starting to get dry. They don't like that either. But the dry can't be all of it, because if it cools down, you'll see a peak in fungi again. But there's something about hot temperatures that are doing it. Although not a lot of beetles like to fly at 100. But they do. Mm-hmm.
You mentioned that one of the advantages of having a symbiotic relationship is that these smaller genomes are able to evolve quicker, and then the host has access to this larger repertoire of genetic traits that might be selectively advantageous.
Right.
So as things warm up, I guess my assumption would have been naively that I would anticipate that the fungus would evolve thermotolerance faster and would evolve to the increasing temperatures and
we don't know if the fungi will do that. With the bacteria, they lose so much of their genome that they don't have the material left to do that.
But can you do those experiments on a Petri plate?
Part of this modeling one of course, the first submission NSF was rejected. And they said, well, you should have an artificial evolution experiment in there. So we added it in, where we tried to force these fungi to evolve by growing them, subculturing, growing, subculturing, sectoring, everything we could do for 2.5 years.
Sounds like it didn't work.
Every few days. And they wouldn't do a thing. I mean, there was no budge. And there's all sorts of problems with growing fungi at high temperatures as well. And then also knowing, I don't think we knew what we were doing either. Because after we did it, I talked to people and said, oh, you should have been doing this and doing this fragmentation thing, and we were just subculturing whatever looked like it grew faster on the culture. But we couldn't get any change at all. We finally just gave up. Maybe that's not long enough either.
So I think I've heard that like leafcutter ants have the symbiotic relationship with a fungus, but that it's actually bigger than just those two, right? There's the symbiotic bacteria on the fungus. So, I mean, what role do bacteria play here?
Well, they don't appear to have any of those like protection bacteria. There are a bunch of actinomycetes that you can culture them and they have effects on fungi, but they're also really bad for the good fungi too, and they're really not, they're more incidental. But for bark beetles, it appears that the role of bacteria that's most important, although this hasn't been studied very much, are some gut bacteria that help them detoxify the terpenes in the trees. And so, you know, it appears that they've got this whole repertoire of gut bacteria that break down the tree defenses. And so I would, from what's been done as far as looking for bacteria on it, outside what they might do, the gut bacteria, that seems to be the only thing that stood out so far, which is pretty important, pretty cool.
How quickly does the fungus like spread in the tree once it?
It's really slow at the beginning. So these are slightly pathogenic. People used to think they were used by the beetles to kill the trees, but that's not the case. But they're probably just virulent enough to hang in this defensive host. So they get inoculated, they sort of sit there for a while. Beetles are tunneling away. And once the oxygen content starts to rise a little from the tunneling and from the tree starting to decline, then they take off fast. So it's not a linear relationship. It's pretty slow at the beginning, and then after a few weeks, they take off as soon as the tree starts to decline. And then they colonize pretty quickly. You know, by the time you go into winter, they're pretty much grown as much as they're going to grow. Pretty close. Mm-hmm.
We had a couple of really severe cold snaps this winter. Is there any hope that that would have any effect on beetle?
Reporters from Bozeman called me and said, hey.
Yeah, I guess so. Is there any legitimacy to this, or is it just?
You know, I don't know what to expect from this winter, because we had a cold snap in October, and nobody thought it was one because it was only 28 degrees. That's not very cold. But the fact is, in October, the beetles seldom are quite ready. They haven't put all the antifreeze in yet. And so I went out to a number of sites by the big hole and stuff, and there was a ton of beetles that got frozen by that. Then the ones that survived, they can survive, you know, minus 30 or so. So you need a really bad cold snap. And our cold snaps didn't get that bad. But what makes me think, and I don't know about Bozeman, but in Missoula in January, we had this like 50-degree weather for like a long time. These beetles don't diapause. When it warms up a bit, they start feeding and developing, and when they do that, they dump antifreeze. And so if they had warmed up enough to start doing that, the next cold snap that happened may have nailed them. So it's actually a very complex thing to figure that out. And there's some cool models that, like the bark beetle one with a couple of our fungal models too, you can use that with the temperature data set to figure out, and some observations in the field on how long the larval tunnels are and all that, to figure out which one of those did it. So I think this summer will be fun to go out and do some tree CSI to figure out if they did get killed, which one of those did it.
So we're about to fund a colleague in Montana, Kurt Stilling. Can you talk about what his contribution to the project is?
Transcriptomics. Out of my range. He's working with these fungi to look if they make some, it's sort of bioprospecting to look if they make some chemical compounds that are useful against all sorts of things. things, actually. But I'm,
Developing a patent?
developing a patent, yeah, so that if these compounds are effective in controlling other fungi, particularly, and so forth. But fungi make all sorts of stuff, so it'll be interesting. He has he has, 3 or 4 compounds that are effective, but only one that's really strong right now. But that's strong. Yeah. But he's just getting off the ground with that, right?
Yeah.
Because we were just
We just,
yeah.
one last question.
Mm-hmm.
Do you have any ideas of different fungal strains that are having any effect on subsequent wood-boring beetle communities?
No idea. So who comes in after,
Yeah.
like Symbiosis and things like that? I don't know. I would imagine it's going to have an impact because these things, just from some studies they're doing in China where they've looked to see what happens to the wood after various ones of these bark beetle-associated fungi have been in. Some of them just deplete the sugar like you wouldn't believe. Some deplete other things. And so I would imagine, not knowing much about Symbiosis nutrition, but it's going to change the substrate use, I would think. So
Based on the strain of the fungus?
I would think it would be different between them, yeah. Some of these fungi, some of the free-living ones, they're in the same genus. They can even be closely related to some of the bark beetle ones. The bark beetle one will concentrate nitrogen, the other one won't. And actually acts as an antagonist. If it's in there, the bark beetles starve to death. So they're going to affect the wood really differently. Might be kind of cool to manipulate that if you could. I never thought about that.
The deadwood cerambicids have a gut flora that includes nitrogen-fixing bacteria.
Cool. Vanilla or something?
I don't actually remember what it is, but
Yeah.
they actually can cause the log nitrogen content to dramatically go up
Wow.
as it rots. So these things do a lot of cool stuff.
Yeah.
Following in on
It would
that.
be
But getting
interesting.
the oxygen in there is a critically important initial
Yeah.
step.
Yeah. That's important for the bark beetles, for their fungi. Neither one do that good. Beetles do better with low oxygen than the fungi.